lectures and then hands-on exercises in the breakout sessions. So we're going to start off with Gloria giving us an intro to the Genome browser. So I'll switch it over to you, Gloria.
Thank you, Omar. Are you able to see my screen?
Yes, looks good.
Okay, thank you. Well, hello, everybody, and welcome to the second day of the ViewPath workshop. Today, we're going to get started with the Genome browser. I'm going to do my demonstration using VectorBase. For those of you that work with parasites and fungi that maybe are not familiar with VectorBase, I will just quickly say that VectorBase is the site that has different arthropods that for importance, such as Anopheles mosquitoes or the yellow fever mosquitoes. I'm pointing here to the header. We also have sandflies, and in addition to vectors that could have importance for viruses, bacteria, or parasites, we also have ectoparasites such as bedbugs. We also have tsetse flies. We also have model species such as Drosophila melanogaster, the fruit fly. And here we have a snail because we also have an intermediate host, not a vector. And this snail is important for schistosoma disease. Okay, so now I'll get back to the topic, the Genome browser. There are different ways you can access the Genome browser. So from the tools menu right here, you can click on Genome browser, but also as we learned yesterday, when you come to the gene pages, we have different links. Here's one for Genome browser. And also we have these windows here. And you have view in JBrowse Genome browser. And this is a tool that we have not developed, but we are very fortunate to be able to provide it for you. So in different areas here. Also, when you come, it seems like somebody else has the microphone open. Maybe you, Omar, sorry. In
it's
different...
not me, so yeah. we can mute people. We'll check and see who's unmuted, and we'll just mute them. But it wasn't me.
Okay, thank you. Okay, so from the dataset pages, we also have different links, including to the Genome browser. I'm going to move this around a little bit. Okay, so you load from here and click on Genome browser. And this may take a little bit to load depending on how many tracks it has. So let's pretend it already open. And here at the top in the Genome browser, it will tell you, we are looking at Aedes aegypti and the strain, the species that we are looking at. But if you want to change species, you come here to this side and select the one that you want to look at. Sorry. Here in this area is telling me that I'm looking at which version of the genome. So this is assembly number 5. And the number after the underscore will be the chromosome or the super contig or scaffold that I'm looking. So at the top, we always have the chromosomes if the species has chromosomes or the data organizing chromosomes. And later we have the super contigs or scaffolds. And later we have the base pair range. From here, I can look in the search bar. I can type the gene name, the symbol, the gene ID, or this location. But if you're going to type the location, you need to keep this configuration of the number, and the number. You notice we have some gene models in red and some others in blue. And notice we have this little arrow. So these genes in red are in the reverse strand, and these ones are in the forward strand. And you can move around. So what I'm doing is I click on this area here and click, and with my mouse, I drag. And you can do exactly the same motion with these arrows here at the top. And we have these magnifying glasses here to zoom in and zoom out. And the difference with the size is how much magnification I have. So notice with the small one, it's just a little bit, and with the big one, it's just much more. This one here is a highlighter. So I selected, notice now it turns yellow, and I click here in this area of the screen and get selected. Now it has an X, so I unselected like this. if I want to move in the area, but I don't click here, I just want to zoom without highlighting. So I move my mouse a little bit to the top. So notice now it didn't highlight. First, because I didn't select it, and second, because I didn't click here, but in this area where I have the numbers. So as I was saying, you can search for different genes using this bar. So for example, I'm going to look for a gap 0047. And notice as I type, I start to get suggestions. And this is the gene that I want. So I click on it. And now it loads. And this is just because these genes has many different transcripts. And that's why I have all of these for that single gene. Okay. What can I, if I hover with the mouse, I can get information about this track. And with this X, I can also close. So here I can come to select tracks, and I want to see, clear all my filters, and I want to see again the gene models. So notice I have two options here. I have this left panel where I can select what I want, or I can start typing here. So I knew already that in gene models, I was going to be able to find this. But I can also see, okay, I want to look at RNA-seq data. So I want this, the combined RNA-seq plots for all the experiments that we have. And for this species, we have 900, here actually, RNA-seq, 914 tracks. That's a lot. So I can use keywords, I don't know, insecticide or anything that you think will be of interest to you. And for these RNA-seq tracks, for example, you want to say, for example, you can say, I only want reads that map to unique regions in the genome, or I only want the ones that are, I know the strand are either forward or reverse, and you start reading the titles, and you decide which are the ones that you want to load, and you back to browser, and those will load here. And notice, and Omar will explain this in much more detail, how the expression sometimes matches and sometimes it doesn't, the gene models that we have, and he will explain to you how to interpret this and what to do about this if you believe the gene models are not correct. What else? Okay, so the other thing I want to tell you, for example, I loaded this one here, and notice how this is RNA-seq evidence. If I have this color orange, it means that I don't know which strand it belongs to. But if I have the RNA-seq data in either blue or red, it means that this matches the forward strand and the red is matching the reverse strand. What else I can do? I was going to show you with this. I can also open peptide evidence. So I want to say, I want all the mass spectrometry data that we have combined in a single track. Or I can say, I want to see everything we have. So we have 6 different tracks. I can load all of them. And notice with a click here in the top bar, I unselect all of them or select them. Back to browser. And now, for example, for this gene here, we have very good evidence, both with RNA-seq data and with peptides. But I want to know who published this data. I want to read this paper. So I come with the mouse and notice now I have an arrow pointing down. I click on it, about this track, and we have a link to the dataset page. And this data set will give me all the details, again, the paper, who is the author, when we release the data, and links to how to analyze the data in the site. Something important that you may want to know is that maybe you want to download a sequence from the Genome browser from specific region. So the way to do that is probably not that intuitive. And the way to do it is that you need to load a reference sequence, back to browser, and say zoom in to see sequence, because what we want to see is the nucleotides and the amino acids, so that's why it's not displaying anything. And notice methionine in green and stop codons in this kind of purple, the 3 open reading frames forward and reverse. And you can say, okay, I want to download, I don't know, I'm gonna say this area here. Oops, sorry, I clicked in the wrong area. Okay, and I come back here to reference, save track data. So I am going to download the highlighted region, this area here in yellow. And if I want to see it, this is the sequence that I'm going to get. And that's pretty much all the things you need to know about navigation. And something important, something else, is that you can share your view. Let's say you loaded different tracks with transcriptomics and proteomics that you want to share with your lab mates or with your PI, a gene that you're working on and how much evidence there is for it. You can share using this link and they will see exactly the same tracks that you are seeing. That's a very quick intro. Omar, I don't know if I forgot anything to say or we are ready to get started with the exercise.
I don't think so. I mean, I think the bottom line is that the Genome browser has, depending on the resource that you're working on, there are anywhere from just a handful of tracks to hundreds of tracks. And it can get, obviously, a little bit bewildering when you go there and you see all these tracks. And so taking your time, looking at the select track section and seeing how things are organized is quite useful sometimes. So depending on the organism you're looking at. So as Gloria is showing you, you have the various categories and subcategories, and you really can drill down by clicking on each of these until you get to a specific data set that you're interested in looking at.
And remember, always clear all filters, because maybe you're looking at transcriptomics and later you start typing something here and nothing appears. So you need to clear the filters and go back to another category, as Omar is saying. So I think with that.
So Carlos is asking in the chat if there's a way to design primers directly from the browser, and the answer is no. There used to be in the previous version of the Genome browser, there was a plugin that allowed you to design primers, but in this version, it's not available.
Is there any other question, or are we ready to start with the exercise? Okay. So I think with that, we can go. Rachel, can we please go to the breakout rooms?
Sure.
Thank you.
Are you in a room, Jesse?
No.
Could you join me in 10 if you're not assigned to one?
I would be glad to. Let's see what happens here. Rachel?
Yep.
Sam has asked me to join him in room 10, unless you need me to have my own room.
Okay, I don't.
Okay.
I can move you.
Okay.
There we go.
Hi, Rachel. I don't seem to be assigned in any room.
Okay, let me see. I think Zoom is. It looks like Zoom may have randomly assigned some folks to the wrong room, so let me fix that and I'll get you put in a room.
Hmm.
Hi everyone, sorry about that. You should all have a room assignment now. Let me know if you're having trouble joining.
Can't see mine yet.
Okay, let me try, let's see. It looks like you should be assigned to room 7. I think if you click on the breakout rooms button at the bottom of the screen,
Mm-hmm.
you should be able to click join beside room 7.
Yep.
Okay.
I don't see it. Thanks.
You're welcome.